Many of you may recognize the title. This is a paper that we published very recently when the blood hits your brain, the neurotoxicity of extravasated blood. Many of you may recognize that this is a takeoff on the 1996 book authored by Frank Endesick, which was titled When the Air Hits Your Brain. So this is the question for this talk, is what happens when the blood hits your brain. As you can see, there are many constituents in blood that are highly neurotoxic. I show at the bottom of the slide three that cause neuronal death. The LD50s are listed there, as are the blood concentrations of those agents. So you can see that something like hemoglobin, at the concentration that it is in blood, is 1,000 times more lethal than the LD50 in neurons. And the other two compounds, thrombin and free iron, similarly are in excess of the LD50 for neurons. There are many constituents in blood that have a variety of different effects. We reviewed these in the paper. I won't go down this list, but you can see that neuronal death is not the only effect of all of these constituents in blood. Neuroinflammation, cerebral edema, vasospasm, seizures, etc., are all consequences of these numerous constituents of normal blood. Very toxic in many different ways, not just cell death, but in all of these other ways. Now, I remember probably nearly 2 decades ago when Misty first made an appearance. And obviously, we credit Dan Handley with this. I was on study section at the time reviewing these grants. And I think that was really the beginning of the modern era of minimally invasive removal of intracerebral hemorrhage. I don't need to go over the results of the most recent Misty 3 data. They're listed briefly there, and you've heard it in the last several trials. Disappointment, except in terms of a benefit and mortality, at least early mortality. There was the interesting finding of greater ICH reduction, leading to better outcomes if the end of treatment volume was 15 cc's, or if more than 70% of the initial volume was resected. That, as you know, also was supported by minimally invasive trials. I just show you one reasonably recent meta-analysis, this study here of 15 high-quality randomized controlled trials, over 2,000 patients. Bottom line conclusion, select patients with supratentorial ICH treated with minimally invasive surgery, significantly more favorable morbidity and mortality compared to those with other treatments. And we all know that that's not enough to bring all our patients to the operating room, but it provides certainly an interesting and important pathway to improving our management of these patients. Of course, Dr. Pradilla's work with minimally invasive parafascicular surgery, what's obviously characterized as a deficit sparing approach because of its minimally invasive parafascicular pathway to the blood clot. This was an early paper that Dr. Pradilla published, again, encouraging results. There are probably 2 or 3 other papers in the literature supporting the use of MIPS in this indication. Again, encouraging, but not yet definitive, unfortunately. These studies that I've briefly mentioned have led to the ENRICH trial, which you've heard detailed a few minutes ago by Dr. Pradilla. And I'll just point out 3 of the inclusion criteria because they bear on the inclusion criteria that we will be mentioning in a moment. That is inclusion of hemorrhages from different locations, at least until the the more recent adaptation of the trial took hold. As we all know, we've heard that good results in terms of hematoma resection result in high 80s to 90% resection, leaving some hematoma behind. What we often don't talk about is the necrotic debris, the white matter that is also left behind. These agents being pro-inflammatory and likely harmful. So the question becomes, can we do more than simply remove 80 to 90% of the hematoma, and can we improve outcomes if we do more than simply remove the hematoma? The hematoma does lots of things. One of the important things that it does is stimulates phagocytosis. One of the approaches that we're interested in advancing is whether we can enhance phagocytosis to remove the residual blood and remove necrotic debris from the injury and from the surgery. You see that there are many constituents on microglia macrophages that are expressed. You see the CD47 there is a classic "don't eat me" signal that's expressed on erythrocytes. That is the signal that prevents macrophages from consuming or phagocytizing the red blood cells. There's been work that has shown that if you use an antibody to mask CD47, you can accelerate hematoma resolution. This is the work that originated in large part with Richard Keep and has been replicated by other groups. So an interesting approach: mask the "don't eat me" signals, enhance phagocytosis. Another compound that I'll mention is the CD36. This is expressed by macrophages and is a very important receptor for recognizing erythrocytes that need to be phagocytized. And we'll talk a bit more about that in detail. We just referred to a couple of the things in red cells and macrophages that are important. It's not just erythrocytes that are a potential target here, but also the products of hemolysis that include hemoglobin, heme, and many other compounds that are harmful, as we reviewed in the earlier slides. So basically, the problem that we face with blood, an intracerebral hemorrhage, is the induction of inflammation and the induction of oxidative stress from the blood toxicity that we've been referring to briefly. So the question is, can we do something to mitigate this damage, this inflammatory and oxidative injury to the tissues? And so I'll introduce here PPAR gamma. PPAR gamma is a transcription factor that is responsible for the expression of a number of helpful molecules that help to oppose or reduce inflammation and that help to phagocytize erythrocytes and necrotic debris. And so in my review of the literature of the compounds, drugs, and approaches that have been suggested and studied for this purpose of accelerating hematoma resolution and reducing inflammation from the intracerebral hemorrhage, the PPAR gamma agonists, in my view, are the most promising because of their pleiotropic effects, including biasing macrophages toward a more prophagocytic M2-like phenotype that will be more phagocytic. Their anti-inflammatory properties, inhibiting NF-kappa B activation, which is responsible, of course, for many of the pro-inflammatory cytokines and chemokines that are activated following a hemorrhage, and for decreasing oxidative stress. So PPAR gamma is responsible, as I say, is a transcription factor responsible for upregulating catalase and copper zinc superoxide dismutase, as well as CD36. The CD36 that we mentioned is necessary in macrophages to accelerate hematoma resolution or clearance, and the catalase and SOD, superoxide dismutase, responsible for reactive oxygen species and for NF-kappa B activation. So if we can hit all of those birds with a one stone of a PPAR gamma agonist, then that would be very desirable. Now, I take no credit for developing this concept. This was developed initially by Jarek Yaronovsky in Houston, who published this paper in the Annals of Neurology many years ago, 2007, where he showed that in a mouse model of intracerebral hemorrhage, administering the PPAR gamma agonist rosiglitazone accelerated the resolution of hematoma over the course of a couple of weeks and improved the neurologic function in this animal model. Now, Jarek and his colleague went on to devise and implement a phase 1/phase 2 clinical trial in humans with ICH, where they administered a different PPAR gamma agonist, pioglitazone, instead of rosiglitazone, for a number of reasons that we don't really need to get into, but it is advantageous. And they studied the safety, and they did a maximum tolerated dose study, again, of rosiglitazone in patients with ICH. Now, rosiglitazone, I want to remind you, is an FDA-approved drug that is used to treat adult onset diabetes, and it acts by enhancing insulin sensitivity in various cells, adipocytes, and so forth. But it also has these other effects that we spoke about in the previous slide. And of course, it's for that, not the relation to diabetes or the relation to insulin sensitivity that renders it efficacious. In any case, in that clinical trial that they devised called SHRINK, as you can see there, this was in ICH patients. No blood was removed from these patients. 42 patients in each group. They discovered and published that the maximum tolerated dose was 1 milligram per kilogram per day. And importantly, there was no safety concern. The reason I bring up the relationship with diabetes is that one might be concerned that this could have a side effect of inducing hypoglycemia, but in fact, they did not see that in their study. So it was safe and it was well tolerated. This study with 42 patients per arm was not powered for efficacy, but in any case, this was the result. Now, unfortunately, the safety part of the study has not yet been published. I've personally reviewed a draft of the manuscript, but I don't know whether they will publish it or not. The only part that has been studied is the maximum. I mean, the only part that's been published is the maximum tolerated dose part of the study. So that gets me to what we are calling now ENRICH Plus. So when I think about this disease of ICH that we have gone through 2 decades now of seemingly being on the edge of helping people, but not being able to get over that hurdle, I wonder if adding something such as a PPAR gamma agonist might get us over that hurdle. Now, again, I don't want to take credit necessarily for that idea. This has been in the literature, others suggesting various compounds, not necessarily pioglitazone, but various compounds to enhance the outcome in patients following minimally invasive surgery. But this is what we're considering now, putting together, is a pilot study, open label, non-randomized, only 20 subjects in this initial conceptualization. And it would be patients with basal ganglia hemorrhage who we think are most likely to benefit rather than, for example, patients with thalamic hemorrhages who are a bit more of a challenge. The way we're devising this, it would have precisely the same inclusion and exclusion criteria as in the ENRICH trial itself. And it would also have the exclusion criteria of shrink for pioglitazone. And that's not much of an addition. It's, hypersensitivity to the drug, certain forms of diabetes and so forth. And the trial is sponsored by NICO. And the analysis would be as follows. The idea would be that we would identify in the ENRICH trial 20 patients who have had minimally invasive or MIPS surgery and who are matched to those in the ENRICH Plus trial based on age, baseline NIHSS, hematoma size and side, using Euclidean matching, using Tom Kent's PPAR's algorithm, Tom Kent, neurologist in Houston. And we would match 20 and 20 patients who have been treated the same way, except for the ENRICH patients, of course, not having pioglitazone. And we would look at the primary outcome that is being examined in ENRICH, and that is MRS at 180 days. We would also look at residual clot at day 7 and perihematoma edema at day 7, anticipating a potential effect of drug on both of those parameters. That's how we envision this pilot study. It's still in the early phases. We have recently applied for from the FDA for IND, investigational new drug status for pioglitazone for this indication. And so we haven't begun it yet, but we hope that we would be able to do so soon. So when blood hits your brain, we'd like folks to think about ENRICH plus shrink equaling ENRICH plus. And I'll remind people in the audience of things that generally as surgeons, we don't operate on tumors without some adjunctive therapy, at the very least dexamethasone for edema and so forth. But we don't have that culture, that habit in ICH surgery. Maybe we should have that. And I would suggest that pioglitazone might be a good way to start. That's it. Thank you. Any questions? I'll be happy to entertain.
Thank you. Again, adding to the list of neurosurgeon clinical investigators trying to push the limits. So fantastic. Pioglitazone is given oral, correct?
Yes.
So what's the rationale between that as opposed to applying it sort of directly in the cavity when you have that opportunity?
Great question. Two-part answer. One is getting FDA approval for intracavity application of a drug versus an already FDA approved for a new indication, a lot simpler. Secondly, the metabolites of pioglitazone are also active, and that metabolism occurs in the liver and, of course, wouldn't occur if it was directly applied in the brain. And thirdly, I said 2, and I'll come up with 3 while I'm talking. The diffusion distance within the cavity, we know from other work, is fairly limited, and we think that in the surrounding area where you have edema and blood-brain barrier opening, access to those tissues would be better with the systemic administration.
Yeah, great. Makes sense.
Yeah.
Anyone else? Comments, questions? Yes.
I don't know if this has been addressed in the previous studies, but there was some studies on chelation therapy and has not shown to be clinically beneficial, but using that as an adjunct to MIP procedure, have you thought about that?
Yeah, I think if I heard you correctly, you're referring to desferoxamine chelation. Is that what you said?
Exactly.
Yeah. Well, that's of course targeting one agent, which is iron, which is released. But there are a lot of other things that I showed you in one of those slides on the way to getting to iron that are still a problem. And by phagocytizing the erythrocytes before they lyse, you prevent the release of iron. So you've done already what desferoxamine does, but you also reduce the inflammatory effect of the other agents as well as the extravasated red cells themselves. So I think upstream of where desferoxamine is working is a better bet and has a broader likelihood of success.